Good afternoon. I am João Negrao. I am from São Paulo University in Pirassununga, Brazil. I would like to thank Dr. Hernandez and Dr. Casey for the invitation for this conference. I would also like to thank the workshop team for organizing this presentation. Thank you very much. I will present the study, the effect of environmental stressors and cortisol realized on milk yield and apoptosis in mammary gland and placenta, in vivo and in vitro response. The objective of this presentation is to report and discuss our results concerning the stress effect on gene expression in the placenta and mammary gland. We have organized our presentation as follows: introduction, general remarks, experimental design to explain our background, results in vivo to discuss the effect of environmental stress on gene expression in the placenta and mammary gland and milk yield. Results in vitro to discuss the effect on mammary epithelial cells, and conclusion. Here, in the introduction, we want to argue that, in general, dairy animals are subjected to several environmental and management challenges that can cause stress during the same lactation. In fact, different stressors are associated with a decrease in milk yield. However, physiological response to environmental challenges can promote animal adaptation. We expect that environmental stress can promote animal adaptation. By other side, global warming can be an additional challenge to dairy animals. It's known that cortisol induces mammary development during late gestation, and that cortisol is fundamental for epithelial cells to start milk synthesis and early lactation. Furthermore, cortisol realized by suckling and milking is important to maintain milk synthesis and lactation. In this context, our research addresses the hypothesis that acute stress, limited exposure to environmental stress doesn't change the mammary gland's responsiveness. However, chronic or cumulative exposure to environmental stress increases the apoptosis rate in mammary epithelial cells. In our studies, we have taken sun and goats as in vivo model and primary mammary epithelial cells as in vitro model. Several laboratory analyses were performed for the purpose to determine the physiological response, milk yield, milk quality, histology and immunohistology, hormone concentration, and gene expression. In our studies, we haven't taken the duration of the stressor to classify acute and chronic stress. In fact, acute stress is established when the stressor happens over a few minutes or hours, and cortisol returns to basal levels between 4 and 6 hours after the stress ends. In contrast, chronic stress is established when a continuous or cumulative stressor is maintained. In that case, the cortisol concentrations remain significantly higher than the basal concentrations for days or weeks, causing negative effects on animals. Here, our goal in this study was to test the effect of different acute stressors on cortisol release and milk yield. This study was performed with 20 lactating goats. The goats were subject to 6 challenges: bath change, vaccination vermifugation, solar radiation, hoof timing, ACTH, and placebo administration. The treatments were performed on mid and lactation. As shown in figure 1, blood samples were collected before, during, and after challenge. As attempt, goat from placebo challenge showed a basal concentration of cortisol. Indeed, different stressors with different durations and intensity showed a different peak cortisol concentration. However, cortisol profiles were similar, with a peak over minutes and cortisol returned to basal concentration 6 hours after imposition of acute stress. Our results concerning the different environmental acute stress on milk yield are presented here in figure 2. In this case, the 6-punctual challenge did not significantly affect milk yield when compared to control group. The negative effect of acute stress on mammary gland is overestimated during the lactation. In this study, our goal was evaluate the effect of immediate cortisol increases on mammary glands and the milk yield. As previously reported, the ACTH dose administered was done to stimulate homogeneous and physiological stress. This study was performed with 30 sunning goats, 2 treatments, ACTH or placebo administration. The treatments and sampling were performed on early, mid, and end lactation. This stress effect was checked by significant cortisol release. As shown in figure 3, the ACTH administration didn't change milk yield when compared to control group. In the same study, mammary biopsies were done on 108 days of lactation. From 22 genes studied, our results show that ACTH via cortisol increased the expression of HSP17 and GHR and prolactin R genes in mammary tissue. In the figure 4, we can see that the administration of ACTH didn't change cell proliferation and apoptosis rate when compared to placebo group. In another study, our goal was to study the effect of different acute stressors on milk yield. In this case, the study was performed with 20 sunning goats, and the goats were subject to 2 treatments, challenge and control. 4 challenges were performed on end lactation. The 4 challenges were solar radiation, hoof trimming, vaccination, vermifugation, and ACTH administration. Control goats were not subject to challenge and maintained in comfort conditions. In Figure 5, we can see that the different cumulative stressors decrease milk yield during and after the imposition of these acute stressors. In fact, the imposition of different stresses sequentially and daily decreased milk yield. In this study, we used the heat stress as a model of chronic stress. The study was performed with 30 sunning goats at late gestation. These goats were subjected to 2 treatments, heat stress in climatic chamber and thermoneutral treatment in a cooling system. In this case, heat stress was imposed during the late gestation, during the last 60 days of the gestation. Mammary biopsies were performed on gestation and lactation. Cortisol, rectal temperature, respiratory rate, and THI were checked during all experimentation. In this study, placenta was taken at parturition. In placenta, heat stress increased the expression of HSP17 and GR and decreased the expression of HST at parturition. In Figure 6, as shown, we can see that heat stress in late gestation also significantly decreased milk yield on subsequent lactation during several weeks. In the same study, heat stress versus thermoneutral conditions from 22 genes studied in the mammary sample, as shown in Figure 1, only prolactin receptor and HSP17 were significantly influenced by heat stress. In summary, acute stress did not have a significant effect on milk quality and milk yield. Cumulative or chronic stress negatively changed milk yield. In this case, here, our objective was to study in vitro the effect of chronic stress on mammary epithelial cells apoptosis. This study was performed with 13 lactating goats. These goats were subjected to ACTH or placebo administration on early, mid, and end lactation. Mammary biopsies were done on day 108 of lactation, 1 hour after ACTH or placebo administration. You used 14 goats, 7 goats from placebo and 7 goats from ACTH treatment groups. These goats were distributed to homogeneous milk production. In this case, the mammary epithelial cells from 14 goats were subjected to 4 treatments in vitro, 0, 10, 100, and 1,000 microgram by milliliter of cortisol hydrocortisone. 10 microgram of cortisol was used to maintain a normal cell culture. 100 microgram of cortisol was used to mimic a low cortisol concentration in plasma, and 1,000 micrograms of cortisol was used to mimic a high cortisol concentration in plasma, like a chronic stress. The primary mammary epithelial cells were cultured for 5 days to mimic the high cortisol concentration like chronic stress. Here, we can summarize our biopsies and the procedure to cell culture. Mammary tissue obtained by biopsies from 14 goats were washed, cut, and placed in individual plates with a basal median. 1 day after plate, cell formed a monolayer with some epithelial morphology. 2 days after planting, epithelial cells are grown, and epithelial cells formed some lumen-like structures. 4 days after planting, fibroblasts were removed, and epithelial cells are grown with typical epithelial morphology to form a confluent culture. The cells from each experimental goats were cultured with basal median and subject to cortisol addition. In this case, there was no effect of previous treatment, ACTH, or placebo in cell culture. In figure 8, we can see that a higher amount of cortisol added in vitro upregulated the GHR, prolactin, and BAX genes, and down-regulated IGF-1 and insulin receptor gene. We can see that this effect was more significantly at 100 treatment of cortisol. Finally, in figure 9, we can see that the addition of cortisol in vitro significantly increases the apoptosis rate and decreases the cell number in culture. This confirms that chronic stress causes apoptosis in vitro. In summary, acute stress did not change the mammary gland's responsiveness. However, chronic stress via cortisol addition in vitro increases apoptosis rate in mammary epithelial cells. In conclusion, chronic stress in vitro changed the expression of GR, prolactin receptor, growth hormone receptor, insulin receptor, IGF-1 receptor, and BAX genes, and increased apoptosis among mammary cells. Our perspectives. Near term, understand how environmental and management factors affect milk yield. It's necessary to make adjustments to dairy farms to improve animal welfare, and milk yield. For the future, understand how stress modulates mammary development and milk synthesis can be an important goal to select dairy animals with lactation persistence adapted to tropical conditions. Further studies are necessary to understand how cortisol modulates the molecular elements of milk synthesis and apoptosis pathway in epithelial cells. This was the papers that support our presentation. We thank the team that worked hard to execute all these studies in the laboratory of lactation physiology. Thank you very much for the research, laboratory assistance, and personnel of animal management, postdoc students, PhD students, and master science students. Thank you very much.